in order to talk about how you actually hear, we need to look at the actual structure of the internal ear, okay, the inner ear. And you have three parts that make up the inner ear, okay? You have the cochlea, okay, the semicircular canal, and the vestibule, okay? And in the vestibule, you have two little sacs. They're called the utricle and the saccule, okay? And the utricle and the saccule and then the semicircular canals are used for balance, okay? So those are the organs that you use to, you know, when you are dizzy, that's what's having a problem, okay? They allow you to have balance. The cochlea is the actual, like, organ of hearing that allows you to hear, okay? So if you look at the picture right here. Okay, so this is the vestibule right here, okay? And then these are your semicircular canals. You have three of them, okay? There's one that goes vertically, or two that go vertically, and then one that goes horizontally, all right? And then this is your vestibule. Notice that at the ends of these semicircular canals, where they actually join with the vestibule, they have these swellings right here at the end that are called ampulla, okay? And we'll talk more about the structure of those and next time, okay? But today we're really going to be looking at the cochlea and the structure of the cochlea and how you actually hear. Okay. So, in the cochlea, so the cochlea is that spiral-looking thing, right? It looks like a snail. If you were to take that lovely little snail-looking thing and you were to unroll it, okay, and then chop it in half, you would see that there's three tubes inside of that cochlea, okay? So, let's draw a picture. So if this was your cochlea, right, that you've now cut in half, you have like three tubes in here, basically. You've got the cochlear duct, okay, the scala, the stibuli, and below, the scala tympani. Okay. These are all filled with fluid. Okay, so your inner ear is actually full of fluid. The cochlear duct is full of a fluid that we call endolymph. Okay, and then the scala tympani and the scala stibuli are called perilymph. Okay. So it's all full of fluid. Inside of the cochlear duct, okay, in the middle, you have this special organ called the spiral organ or the organ of Corti, and that is the actual place where the hair cells that pick up the sound that you're hearing right now live. Okay, it's in this little cochlear duct, okay, in the spiral organ or organ of Corti, same thing. Okay, and so that's your organ of Corti / spiral organ. All right, make sense how this is structured right now? Yeah? All right, so these three tubes are completely separate, right, by these membranes. And so it's basically like one tube that has three inside of it, and they're all completely separated by membranes. The scala tympani and the scala vestibuli are completely separate, except for at the very end of the cochlea. Okay, so again, if you were to unroll that cochlea and it'd just be like laying out flat, okay, you'd have the scala vestibuli on the top and the scala vestibuli or tympani on the bottom, and then those two would meet at the very end of the cochlea. Does that make sense? So they join together at the end of the cochlea. Yeah? Okay. So they meet at the end. Okay. So the spiral organ, which is the actual organ that allows you to hear, also called the organ of Corti, right? That sits on what we call the basilar membrane. Okay, the basilar membrane is this membrane right here. Okay, so this membrane is called the basilar membrane. All right, this one right here. Okay, and then your actual organ of Corti, spiral organ sits on top of it. Okay. And inside of that spiral organ, or the organ of Corti, you have a couple different kinds of cells. You have supporting cells, and then you also have the actual hair cells that pick up the sound waves. Okay, so those hair cells are responsible for you hearing right now. So the hair cells sit surrounded by supporting cells, and they stick their lovely little hair cells up into the endolimb. Okay, so here's your spiral organ. Inside here, you would actually have little cells that are hair cells that stick up. Okay, here, and then those actual like hair cells, the hair cells, the hair of the cells itself actually stick into another membrane. It's called the tectorial membrane. Does that make sense? Okay, so this whole thing is the spiral organ, or the organ of Corti. Okay, in here you have hair cells. Okay, and they're called hair cells because they have little projections that look like hairs that stick off of them. And then those hair cells are embedded into another membrane that sits on top of it that's called the tectorial membrane. Does that make sense? Yes? Yes, this is all inside of your cochlea. Okay, so in the spiral organ, okay, so here, this is the actual spiral organ itself, okay? These are your little hair cells, the hair cells that are going to allow you to hear, and then this is the membrane that the hair cells stick into right here, called the tectorial membrane, okay? And so then you've got all of these other supporting cells around it, right? And then here's your little hair cell too, and then that hair cell is connected to right here, which is a neuron, okay? So here is a better drawing than I could do of the inside of your cochlea, okay? So up here, this is the scala vestibuli. All right. This is the cochlear duct. This is the spiral organ. Okay. And then this right here, that would be the scala tympani. Right, and then here's your basilar membrane, etc. Okay, so that's what we're looking at. And then these little things are your hair cells, and then here's your tectorial membrane. All right? So does the structure of the ear, the cochlea, make sense? Yes? Okay. So the other couple things that you need to remember is that the scala tympani, or sorry, the scala vestibuli up here, it has, where is it? The oval window, sorry, right here. Okay, so this is the oval window right here. The oval window connects to the scala vestibuli, which is the upper tube. All right, and then on the lower tube, okay, which is the scala tympani, okay, you have the round window. All right, and if we look back at this picture, you can see it. So this is the round window right here, and then here's your oval window, where the stirrup or the stapes connects. All right, and so this one connects with the upper tube, and then this one connects with the lower tube. Does that make sense? Okay. So here we go. How do you actually hear? So sound waves travel through air, okay? And they're moving through the air. Your auricle, or the outside of your ear, part of the ear that you actually see, it helps to kind of a little bit funnel that your auditory canal, your external auditory canal. Okay? Sound waves travel through that external auditory canal, hit the tympanic membrane, your eardrum, and causes it to vibrate. Okay, so your tympanic membrane moves back and forth. When that tympanic membrane moves back and forth, it moves those three bones. Okay, the hammer, the angle, and the shady. Yeah. Okay, so it moves those three bones, and those three bones start to vibrate too. Okay, based on how fast that eardrum is vibrates those bones. Remember that stirrup sits on the oval window of your cochlea, right? So when that stirrup starts to vibrate, it's pushing on the round or oval window of your cochlea, and it changes those air waves, like the sound waves in the air, to pressure waves inside the liquid that fills up the cochlea. Does that make sense? Yes? Okay. So, as soon as the pressure waves hit in the cochlea, they start to move, okay, through this liquid. And so when they start to move through this liquid, they hit on here, sorry. Okay, so sound waves are moving through here, okay, as pressure waves now. And they'll hit this membrane right here. Okay, when they hit this membrane, it causes the liquid in here to move, okay, which pushes down on the tectorial membrane, which pushes down on the hair cells. Does that make sense? And when it pushes down on the hair cells move, okay, which causes them to release a neurotransmitter, which communicates with the neuron, which sends a signal to your brain that you're hearing. Does that make sense? Okay, so sound comes through the auricle, right, through this external auditory canal, hits the tympanic membrane, this vibrates, moves these three bones, okay? This bone moves back and forth, okay, which makes pressure waves in the perilymph, in the scala vestibuli, okay, which hits the vestibular membrane, which causes pressure waves in the endolymph of the cochlear duct, pushes on the tectorial membrane, which pushes on the hair cells, okay? And then those hair cells release neurotransmitters and send the signal back to your brain that you're hearing, okay? You've created pressure waves now in this cochlea, right? And pressure waves, if you were to just perpetually have more and more pressure waves build up as you heard more and more, eventually your cochlea would burst, right? Because you just get all these pressure waves in there and they couldn't have any place to get out. So actually, you have the pressure waves that move through the scala vestibuli all the way around through your cochlea. And remember, the scala vestibuli and the scala tympani meet here at the end, okay? And so the sound waves travel all the way through, they get to the end, and then they go through the scala tympani all the way back out, and then the pressure gets released at the round window on the cochlea. So the pressure comes out through the round window. Does that make sense? So it goes like all the way through the cochlea, and then it comes all the way back, and then the pressure gets released at the round window. Okay. So basically, how do you hear different like pitches? So like high versus low, or like how loud stuff is. Frequency, basically. Really, so the membrane and the cochlear duct kind of up here closer to the beginning of the cochlea, like up here. This is actually where you sense or hear higher sounds. Okay, and then when you get down here towards the end of the cochlea, that's actually where you hear lower sounds. The loudness of the sound is based on frequency. So how often is this getting hit? Okay, so you have certain hair cells that are here, and if they get hit or stimulated, your brain interprets it as high sound. If they hit back here, your brain hears it, bless you, as a low sound. Okay, loudness is how often they get stimulated. So the louder it is, the more they get hit, and the more they send the signal to your brain, which gets interpreted as something is really, really loud.